Hello viewers, I am Sameen Khan, a student of MSc Zoology, Semester 4, RD and DJ College, Munger. As a part of our curriculum, we have been asked to prepare and submit an assignment. The topic assigned to me was hypothalamic neurotransmitters. Before starting, I would like to express my heartfelt gratitude to our revered teachers, Dr. Mayank Madhukar, head of the Department of Zoology, and our assistant professor, Sri Saurabh Yashasvi, without whose active support, encouragement, and teaching ability par excellence, completion of this presentation would not have been possible. Now, let us have a glance at the topics covered under this presentation. Without any further ado, let us now come down to a brief introduction of the topic. Hypothalamus is the integral region of the brain lying below the thalamus and making up the floor of the third cerebral ventricle. It is a small, cone shaped structure that projects downwards from the brain, ending in the pituitary stalk, that is, a tubular connection to the master gland. The hypothalamus and the pituitary gland are connected by both nervous and chemical pathways. The posterior portion of the hypothalamus is called median eminence that contains unmyelinated neurons, and its main function is to act as a gateway for the release of hypothalamic hormones like thyrotropin releasing hormone, luteinizing hormone releasing hormone, corticotropin releasing factor, growth hormone releasing factor, prolactin inhibiting factor. And the target tissue of the above mentioned hormones is that of the anterior pituitary. The next subtopic is hypothalamic regulation of hormone secretion. The many interconnecting neurons of hypothalamus are nourished by a rich supply of blood, which carry the released hormones as well. But the stimulus for secretion and formation occurs through neurotransmitters all through the brain, and it is a process by which one neuron communicates with another via a synapse, that is, a small gap between the nerve terminals. These may be called as presynaptic or postsynaptic in reference to the direction in which an impulse is traveling, with the presynaptic neuron transmitting an impulse to the postsynaptic neuron. Transmission of the impulses need a chemical substance that diffuses across the synapse from the presynaptic membrane of one neuron to the postsynaptic of another, and that substance is called a neurotransmitter. The neurotransmitters are synthesized and secreted in the same manner as the synthesis of any protein hormone, with the exception that the neurotransmitters are contained within the neurosecretory granules that are produced in the cell body and migrate through the axon to the nerve terminal from which they are discharged into the synaptic space. The peptides are packed in dense core vesicles which are released by exocytosis. Its mechanism is similar to that in other cells and require an increase in the intracellular calcium ion concentration. This secretion plays a widespread role in the organization of the central nervous system. Some of the classic hypothalamic neurotransmitters are epinephrine, norepinephrine, serotonin, acetylcholine, dopamine, glutamate, gamma aminobutyric acid, etc. A large number of additional neurotransmitters have also been discovered, which are called as neuropeptides. These neuropeptides may function as neuromodulators, whose main role is to increase or decrease the action of neurotransmitters. Example of neuromodulator is opioid, like enkephalin. Role of neurotransmitters in neurosecretion. Some of the hypothalamic neurohormones are called as releasing hormones because their major function is to stimulate the secretion of hormones originating in the anterior pituitary gland. For example, luteinizing hormone releasing hormone acts on the anterior part of the pituitary gland to release luteinizing hormone. Similarly, thyroid releasing hormone acts to release the thyroid stimulating hormone. Apart from this, the neurotransmitters also exhibit inhibiting action. The hypothalamic hormone somatostatin has primarily inhibiting action on the growth hormone secretion, although it can also inhibit the secretion of other hormones. Another hormone, dopamine, has an inhibitory action on prolactin, formation and storage of neurohormones. The cell bodies or neurons that produce these are not evenly distributed throughout the hypothalamus. Instead, they are grouped together in paired clusters of cell bodies called nuclei, which can be easily identified as the Nissl body that is not scattered evenly throughout the cytoplasm of the cell. Rather, it is collected at the periphery so that its nucleus can be seen easily. Vasopressin and oxytocin are produced in the hypothalamus where they are packed into neurosecretory granules, and in response to a signal, these are extruded into a capillary network that feeds directly to the general circulation. The extrusion might take place from the bulbous enlargements serving as loci for the release of neurohormones. On the basis of the above mentioned facts, I have come to the conclusion part of this presentation that the secretion of specific neurotransmitters and neuropeptides leads to an organized and directed function of the overall central nervous system and the endocrine glands, out of which hypothalamus is an important portion. It produces and secretes not only neurotransmitters and neuropeptides, but also several neurohormones that alter the pituitary function. The neurons producing and secreting neurohormones are the true endocrine cells, as the hormones are incorporated into secretory granules which are carried through the axon and stored in the nerve terminals located in the posterior pituitary gland. In response to a neural stimuli by the neurotransmitters, the contents of the secretory granules are extruded from nerve terminals into the capillary network. Here, in case of pituitary gland, the secretory granules are carried through the hypophyseal portal circulation and delivered directly to the anterior pituitary gland. Thank you.